So hopefully in the second half of this video, we're going to work through some Punnett squares. Those are charts that we use in order to determine what the possible phenotypes and genotypes are for any two-parent combination. So first, let's start by breaking down pretty slowly what we mean when we're trying to determine what to put into a Punnett square, how we need to break down information. So we have parent one on the left. They have a phenotype of a normal thumb. And they have two normal thumb alleles. So they have both of their parents contributed a normal thumb allele. And so they are 100% likely to pass on a normal thumb. So whatever offspring they have, parent one can only pass on normal thumb alleles. Parent two has a hitchhiker thumb phenotype. That is that they have two recessive alleles for this particular gene, and they can only pass on hitchhiker thumbs. So parent one can only pass on normal thumbs alleles, and parent two can only pass on hitchhiker thumb alleles. So what does this mean for the phenotype of the offspring? So over here on the left, we have parent one's two big N alleles, or two normal thumb dominant alleles. And on the top, we have both of parent two's little n or hitchhiker thumb alleles. And so we can take from here one big N, one little n, one big one little n, and so on. They're all going to be the same. And so 100% of the offspring between parent one and parent two are going to have normal thumbs, and they're all going to have this heterozygous one big N, one little n genotype. It's important to note that we're talking about parent one and parent two here, because these are not sex dependent, since these are located on the 22 non-sex chromosomes. It doesn't matter if you get one from mom or one from dad, and we're just going to refer to them as parent one and parent two to prevent confusion. So in this example, parent one is different. So parent one has a normal thumb, but since we know that normal thumb can be achieved in two ways, either by a homozygous with two normal thumb alleles, or heterozygous when one normal thumb and one hitchhiker allele. This parent has one normal thumb, one hitchhiker, heterozygous for this particular allele. And so they're 50% likely to pass on a normal thumb allele and 50% hitchhiker, because it's random. Their kids can get either one of these. And parent two says the same, so they're 100% likely to pass on a hitchhiker allele. Looking at our Punnett square, we have parent one over here, heterozygous. We have homozygous recessive on the top for parent two. And so this first cell gets, the cell meaning this particular part of the table, not an actual cell, gets a big N, little n, and same here. And these two both get little n because they're getting those alleles from each parent. So the progeny have the genotype 50% big N, little n, and 50% little n, little n. So the 50% that have the big N, little n, who are heterozygous, are going to have normal thumbs. They're going to have a normal phenotype. And And the ones on the bottom here who have two little n's or two recessive alleles are going to have hitchhiker thumbs. So the phenotype for the progeny is 50% normal thumbs and 50% hitchhiker thumbs. The genotype is that they are 50% heterozygous for this allele and 50% homozygous for this allele. So diseases can be dominant, like Huntington's disease, and this works much the same way of the example that we just gave. So Huntington disease, which you might be familiar with, is really devastating, and it's a dominant disease, meaning that if you get an allele from either parent, then you are going to be affected. A Huntington's disease allele. So in this example, we've got parent one who's homozygous recessive, so does not have either Huntington's allele. And up here we have a parent with one Huntington's allele and one non-Huntington's allele. And so if we go through our Punnett square, we see that this column is heterozygous. So one big N, one little n, one big N, one little n for Huntington's. And this column all have little n's, and so they are not carrying Huntington's. And so the progeny are going to be 50% heterozygous and 50% homozygous. The 50% that are heterozygous are going to have Huntington's, and the 50% that are not heterozygous, that are homozygous, are not going to have Huntington's. So if someone has this disease, it has an allele for this disease, they're going to have this disease. There's no such thing as carrying the Huntington's allele, because if you have any allele, then that is going to dominate. Let's look at one other example. So if a parent was to inherit two Huntington's alleles from their parents, even if their partner had no Huntington's allele, it is guaranteed that their child would have Huntington's. All of their children would be heterozygous and then have Huntington's because it's a dominant disease. And so this is an example where genetic testing often comes in, and it's very possible that these people would test before that they have children to prevent this disease. It's pretty terrible. People normally die in their 30s or 40s. Diseases can also be recessive, so that means that you need two alleles in order to have the disease. And so since it's a recessive allele, we're going to denote the diseased allele with a little n. And so sickle cell, which you guys are probably familiar with, occurs on the 11th chromosome. It's a single point mutation. It makes a mutant protein that creates blood cells, red blood cells that look like a sickle instead of the normal red blood cells that are nice and round and healthy looking. And so causes a lot of issues down the line. You guys are all familiar. So in this example, parent one has normal red blood cells, but they have the alleles for one red blood cell and one sickle cell. So this is the instance I was talking about a little earlier, that they are a carrier of sickle cell because they do not have sickle cell themselves. Their phenotype is normal red blood cells. However, they can give a sickle cell allele to their offspring. Same with parent 2. Parent 2 is also a carrier of sickle cell. And so they are both 50% likely to pass on a normal red blood cell and 50% likely to pass down a sickle cell allele. They are both heterozygous. And since this is a recessive disease, again, they are both considered carriers because they don't have the phenotype themselves, but they can give it to their offspring. So let's look at two heterozygous parent mating. So this, so big N, we have a homozygous dominant. Big N, little n, we have a homozygous, a heterozygous, big N, little n, and we have a heterozygous, and then we have a homozygous recessive. So if we look at this, this person here, since it's a recessive disease, they will not have the disease. They'll have normal blood cells. These two are both going to be like their parents, carriers of the sickle cell allele, but they will not themselves have sickle cell. And this person is going to have sickle cell. So we have 75% with normal red blood cells and 25% that have sickle cell themselves. And then 50% of the offspring will be carriers. So meaning that they have normal red blood cells, but they still carry the sickle cell allele. And so now we're going to talk about sex-linked diseases. So not autosomal diseases like Huntington's and sickle cell that we've already been talking about. So one really well-known sex-linked disease is color blindness. This is just going to be the example that we're talking about. But first, we're going to think about how DNA passes between generations of people. We talked about this last week. It occurs during meiosis. You have the same thing as mitosis, except some crossover, which leads to genetic diversity. And you end up with these sex cells, which are haploid, meaning that they have half the number of chromosomes as normal cells. And so then two of these nice little sex cells combine in order to make an embryo. And so that's how people end up back with the same number of chromosomes that their parents had. One from mom's DNA, one from dad's DNA. Here's our karyotype again. And again, we can look and see that this karyotype is XX. It has two copies of the X chromosome, and therefore it is a girl. If it was to have one X chromosome, one Y chromosome, it would be a boy. And so there are multiple diseases that occur on the X chromosome. So if there's any kind of variation from normal on the X chromosome, then this is going to be inherited differently than our autosomal diseases that we've talked about. This is because girls have two copies of it. So they have both X chromosomes, one from their mom, one from the dad. And boys only have one copy, only the copy from their mom. And so they don't have the copy from their dad to possibly mask that. And so we see different patterns of inheritance of X-linked diseases. So we're only going to talk about X-linked diseases. There are also Y-linked diseases. Um, and so a question for you to consider is, how likely is it for a woman to get a disease that's located on the Y chromosome? So just think about that for a second. There's no chance of a woman getting a disease on the Y chromosome because they don't inherit a Y chromosome. Um, but males are more likely to get diseases that are located on the X chromosome because they have no other chromosome to compensate for any possible deleterious alterations. So we're going to talk about color blindness. As I said, it's located on the X chromosome and it's recessive. So meaning that you need, if you're a female, 2 copies in order to express the phenotype. But if you're a male, you only need 1 copy for the phenotype because you don't have the other normal allele compensating. So let's look at this example. So we have the dad here, the XY, and we have the mom up here, 2 X chromosomes. And so say the dad, X and Y, this little n, means that the dad is colorblind himself. And the mom is a carrier for color blindness because she has this big N allele that is guiding her actual vision. She's heterozygous. And because of this, she's heterozygous for a recessive disease, then she's considered a carrier. So she doesn't have color blindness herself. The dad does. Let's look at their offspring. So this person, this girl, is getting a big N from mom and a little N from dad, meaning that she is also a carrier. She's also heterozygous, like her mom. This one is getting a little N from both dad and mom. And so she's going to be a female with color blindness. This is really rare. It's like 1 in 10,000 or so. We have mom giving the big N down here to her son. Dad's giving Y, obviously. And so son does not have color blindness, and this son does because he's only getting this colorblind allele from mom. So our final phenotypes are that you have 25% carrier daughters who are unaffected, 25% affected daughters, 25% affected sons right here, and 25% unaffected sons. So hopefully you see the difference between that and the inheritance pattern of autosomal diseases. Let's go through another example. So here, if mom is colorblind, again, pretty rare, but dad is not, then this daughter is going to be heterozygous, meaning she's a carrier. This daughter is going to be heterozygous, meaning she's a carrier. This son can only get a little N allele from mom. That's his only X chromosome allele, and so the only allele that he gets for this trait, and therefore he is colorblind, as is this son. So in this example, the daughters are all heterozygous, so they're all carriers for this disease, and the sons are all affected by color blindness.